Hello, everyone. My name is Mr. Baker. I am starting a new unit today for my biology class. We are going over modern genetics. So, last we talked about Mendelian genetics and some very basic stuff. Now, we're going to cover some of the more recent advancements, and that means we're going back to about 1990 or so and working up from there. So, the past 30 years, give or take, when I'm recording this. And if you are part of my class, obviously there is a link to the unit's vocab in the bottom corner of this presentation. Let's go ahead and get started. The first topic I want to talk about is the Human Genome Project. We're going to be asking the question of what's its purpose, what did it do, what are some benefits that it has gained us, because that was a very big endeavor on an international scale. And then we're going to talk about DNA fingerprinting. And while we are scientists and we usually don't talk about if something is a good thing or not, if it's beneficial, if it's ethically moral, DNA fingerprinting is one of those that we might have to think about it a little. So the Human Genome Project was a international project. It was headed by 10 colleges and universities that dedicated their time to decoding all DNA, and that in humans. So we're specifically talking about humans, and we're identifying the sequences of base pairs. So the C's, the G's, the T's, the A's, right? Cytosine, guanine, thymine, and adenine, right? And what they did was they studied all of them and they cataloged as much as they could what those actually meant, right? And this is the baseline human DNA in the genome, right? And mapping them onto specific chromosomes so that we know what they are supposed to look like. That has given us a huge boon on when those don't quite go as planned. For my class, at least, you don't need to know the dates. I do want you to know that it took about 13 years, so from 1990 to 2003. The image that is below my face is a bookcase that actually has all of the published human genome. And so each of those binders is a 3-inch binder that is full of, hold on, I get a cough, so I'm going to pause the recording. Sorry about that. It's just full of the DNA sequence. So it's just the letters C, G, T, and A. That's all it is. It's DNA. And it is a string. Now, there's also a key to this that actually says, okay, here are which part of which chromosome we're talking about. And you can see that on the ends of those, right? You can see the numbers 2 through 22 in this bookcase, and then at the very bottom, the sex genes, X and Y. If you are looking at it pretty closely, you might notice that X has a lot more books than Y. Y is a shorter chromosome than X. It's actually physically smaller, so it takes up less space. That's true for a lot of the other chromosomes too. They aren't all exactly the same length in printed form because they are all different shapes and sizes. They are all generally about the same, but there's still a bit of difference there. So the project was deemed complete in 2003, and a accuracy study was performed on it over the next few years. They got about 92.1% of all the DNA in the genome, and that was at 99.99% accuracy. So for every 10,000 letters, there might be one that's mistaken. But this is for a genome, right? So this is the average. It's the norm. So there could be mutations. There could be molecular differences. There's a lot of stuff that we've talked about in class, actually, that could be reasons why it's slightly different if you look at it on an individual scale, all right? And they didn't cover specific parts of chromosomes like telomeres, which are chains that are put at the ends of chromosomes that protect them from being deleted in a like frame shift mutation or a stop mutation that could cause a problem with DNA replication. Telomeres, part of what they do is to fix that. Also, in the 1990s to 2003, telomeres weren't widely researched yet at that point in time. So they were like, yeah, those are there, but we're not sure what they do, so we're not going to cover them yet as well. So all of this data has been published. You can actually go on the internet and download it if you want to. It is out there. It has been made readily available to the public. GenBank also uses this to actually say, here are some actual useful information from this giant amount of data. And they've worked to kind of not just decode it, but categorize it and quantify it a little bit better. Right? It has had two big things I want to talk about as benefits that I expect my class to know. This is not a end of Human Genome Project benefit list. This is just a couple examples. All right. So in molecular medicine, that's treating medicine at the very, very tiny level, right? And not just saying, oh, you've got a headache here, take some aspirin, right? But cancer identification and treatment. So cancer is a genetic disease. It affects the DNA of a cell and causes it to mutate and then causes it to replicate itself exponentially. That's why cancer grows so much. Being able to say, hey, that is cancerous, or no, that's not cancerous, a lot of that comes from being able to know what the human genome is supposed to look like. So they compare it to information from the Human Genome Project. We've also been working towards treating of cancers over the past 45, 55 years, give or take. And the information from this project has helped scientists actually determine some treatments for specific cancers. That's something we'll talk about later in the unit, so I'm not going to get too in depth right now. And then microbial genetics. So looking at bacterial diseases and infections has been beneficial in comparing it to the human genome, because if we see a specific string of nitrogenous bases in the DNA of a bacteria, we might be able to say, oh, I think that does blank. Or that's a weakness we might be able to exploit by using X medicine, right? We also have started to engineer specific bacteria that can do certain things for us. One of the big ones right now in environmental protection is trying to make bacteria that will eat plastics so that we can recycle plastics a little better. I mean, yeah, using my non-branded, right, water bottle instead of buying a water bottle every time I want a drink to take with me someplace does conserve plastic, but being able to melt it down and reuse it isn't something that we can do as efficient as we would like to. And having bacteria that can break it down into its constituent parts is going to be a big boon for the environment, especially when we talk about some of the single-use plastics that are out there. Moving on, DNA fingerprinting is another type of genetic research that's been done specifically in the past 30 to 40 years, and it's been used for a lot of different things, including as part of the genome mapping Human Genome Project. There are a few specific steps I would like you to know if you're in one of my classes, and most biology classes are probably going to ask you to know these. You may get to do these depending on what school you're at. So first, a DNA sample is taken. Cheek swab, blood, there's a lot of different ways this can be done. Generally, it's used in liquid form. If it's not a liquid form of DNA, it may have a few steps done to it before this. Right? So we're going to use something called restriction enzymes to cut that DNA into smaller pieces. And so instead of one long string, it's going to be chunked. Right? And we specifically use restriction enzymes that will cut it into different sizes of length. Right? And then we're going to put those into this gel. It is a gel that specifically floats inside a buffer solution. And that buffer is going to be able to conduct electricity, and the gel will conduct electricity as well, so that the DNA segments that are inside these wells, these little holes in the gel, can move based on that current. And because they're going to be attracted to the positive terminal of the electricity, you can see that in the bottom part of the diagram here, the DNA will move through the gel. In that picture, it's left to right, but it's always from the negative towards the positive, right? So from the anode to the cathode, right? And smaller DNA segments, because they don't have as much mass, can move farther and faster than bigger DNA segments, right? Inertia is a thing, and we're going to get some what's called banding visible, right? And while this picture down there is just a diagram, a lot of them do look exactly like this. Your DNA, the DNA sample might be dyed a specific color so that you can see it better on the gel, so that it stands out more. But we can see these bands of where these specific segments moved to across the gel. And we can use that specific banding to identify or match one DNA sample to another DNA sample. Or we can use that information to say this is most likely a familial relationship, so part of the same family. Mother and father and siblings can be identified this way. Usually, you don't have to run a DNA fingerprint for a mother because we know who the kid came from, but paternity tests, this is one of the ways they are done. If it's a adoption or something like that, a DNA fingerprinting might be done to find a mother as well. In the criminal investigation world, they can take a DNA sample found at a scene of a crime and match it to a specific suspect to say, oh, this person is guilty or this person is innocent, or at least this person was there, right? Now, if that picture wasn't great, here are the steps that I've wrote for this. So restriction enzymes are, remember, those are just proteins that have a job, right? Their job is to cut the DNA into segments, so pieces specifically into different lengths, right? Now, something that wasn't in the last image was that we're going to use something called a PCR reaction. That's kind of redundant because the R stands for reaction. We're going to use PCR to replicate those DNA segments so that there's more of them, right? So this is just DNA replication, but forced. So we're going to grow more DNA, right? We're just copying it. We're making more of it. And then we're going to use gel electrophoresis. That's the process I was talking about, to separate those segments based on their size. Remember, the larger segments move less, right? So if I were, say, a teacher, which I am, asking students a question, say on a test, this might be a good essay question example, right? Explain how DNA fingerprinting works. And the goal, of course, is to match DNA samples based on their specific banding patterns from a known to an unknown, or vice versa, or to identify familial relationships. Now, one of the ways that they get it, that right, they being any government or private entity that wants to build a database of DNA samples to use for DNA fingerprinting, that's something that is done. And that may or may not be a good thing. Do you own your DNA? Maybe, but your DNA looks a lot similar to other people's DNA. Do you care if somebody has a copy of your DNA? You might, you may not. Humans shed a lot of DNA every day. Skin cells, hair, spit, everything that comes out of your body has a little bit of DNA in it. Most people don't, make sure that they know where all their skin cells go at the end of the day. So it's very easy to get a DNA sample from someone and being able to match that to something specific may or may not be a good thing. Governments across the world right now are doing multiple different things with DNA fingerprinting and building DNA databases. I'm not going to say if it's a good or bad thing, but I do want you to think about it, right? Is it a beneficial thing to the people that live there? Is it beneficial to everyone? Is it beneficial to only those people who say remain anonymous? But that is your thought for the day, let's say. All right. Keep asking questions and keep learning. Have a good one.